We have to go through 2 divisions, however. Mitosis was just 1 round of division, and we ended up with an exact copy. Meiosis, we're actually dividing and reducing. In fact, that was the old word for meiosis, was reduction and division. Meiosis I, we cut the number of chromosomes in half. We rest for a little bit in a phase called interkinesis. And then depending on what type of cell you are, you may immediately go through Meiosis II, which is a division of chromosomes. So sperm cells go through a reduction, a rest, and a division almost immediately. Egg cells go through a reduction when you're a fetus, and don't go through division until fertilization, much later. But meiosis is just meiosis I, interkinesis, meiosis II. In meiosis I, we replicate the chromosomes. And so each chromosome has those sister chromatids. They're in those X shapes that we saw last chapter, where they duplicated. The big difference in meiosis I, though, is during metaphase, we don't all line up in a straight row. Our homologous pairs line up next to each other. And so chromosome 1 lines up next to chromosome 1, chromosome 2 next to chromosome 2, 3, 4, 5, all the way down. This is called synapsis. And it's one of the best ways to differentiate between mitosis and meiosis. Mitosis, you're all on a straight line. Nobody's lining up next to anybody. In meiosis, you line up next to your pair. You line up so close that you may accidentally share information. You may exchange information inside of your own cell, so things get mixed up. That's one of the reasons that there are 70 trillion different combinations between within your own cells, really, when you have a fertilization event. That's just because you can do some mixing in your own cell, which is wild, but it happens. They align themselves, then we separate. And so each daughter cell receives a duplicated portion. And so it's very similar to mitosis. So we have our chromosomes, and they duplicate. And so we see those X shapes. They line up in a process called synapsis, and then we go through a division. But each cell, even though we have a reduced number of chromosomes, notice we still have too many copies. Those sisters did not separate. And so we have to go through meiosis II to separate those sister chromatids. Between meiosis I and meiosis II, DNA is not replicated. So star that or use your exam highlighter to say DNA is not replicated between meiosis I and meiosis II. Why would you? The goal of meiosis is to reduce your chromosomes, not keep making more copies. If you would duplicate that, you'd end up with too many if the sperm or egg combined and has too many copies. And so between meiosis I and meiosis II, you don't duplicate. In meiosis II, the sisters separate, move to opposite poles, and now you have 4 cells that each have a reduced copy of chromosomes. And so at the end of meiosis, you end up with 4 haploid cells, generally speaking. And so we have meiosis. We went through our meiosis I, we duplicated, we lined up, we split, but we're not done because those sisters are connected. So we go through another round of meiosis where we cut those in half. Now, if you're a sperm cell, you have 4 copies of a reduced individual, a haploid cell. If you see a 1 or a 2 after the phase, you know we're talking about meiosis. Mitosis only goes through 1 round. So we don't number it. Why would you? There's just one round. Meiosis has 2 rounds. So we need to know the difference. So that's a big way that you can help yourself on the exam is if you see a number at the end of a phase, we're talking about meiosis. We don't number mitosis. And so, we've seen these words before, prophase, metaphase, anaphase, telophase, except we stick a 1 at the end, Roman numeral 1. We can rest for a bit in a phase called interkinesis, and then go through the phases again, prophase, metaphase, anaphase, telophase, and then stick a Roman numeral 2 at the end of each one. You're going to see some similarities here. In prophase 1, a spindle forms. The nuclear envelope fragments. Nucleolus disappears. Each chromosome is duplicated. Old news, just like prophase of mitosis. However, our cells begin to line up, and they don't line up in a direct line. The homologous pairs line up. We said they synapsed. Now, when they synapse, we use different terms to talk about our sisters. When we synapse, we can use the word bivalent and tetrad. Bivalent is referring to our 2 homologs. Tetrad is referring to in this situation, there are technically speaking 4 copies of that chromosome. And so bivalent just says we have 2 homologous pairs here. Tetrad is saying, tetra means 4. Tetrad is saying we have 1, 2, 3, 4 copies of that chromosome in meiosis 1. We divide, and our bivalents would separate into their individual cells at the end of meiosis 1. And so it's just new terminology. I don't use bivalent and tetrad a lot, but you'll see it in the lab. I prefer to say homologous pairs. If you take genetics, you'll use bivalent and tetrad. It's not terminology I use, but it is going to come up in the lab. Metaphase 1, I'll say it again, even though you've heard me say it multiple times now at this point, your homologous pairs line up. Do I have a picture? I don't. You already saw a picture. And so instead of being chromosome 1 through 46 in a straight row, you're going to see homologous pairs line up like this at metaphase. That's a big determiner. So if we were talking about the difference between metaphase of mitosis and metaphase 1 of meiosis, this would be a big one. In metaphase 1 of meiosis, homologous pairs line up. They synapse. What can occur when they synapse that increases genetic variation? What are the 2 things that can occur? Crossing over and independent assortment. We don't care which side they're on. We don't care what order they're in, as long as the homologous pairs are next to each other. That's independent assortment. Anaphase 1, they just move apart to opposite poles, but the big one here, the test question that's always asked is, those sisters are not separated. So they separate the poles, they go to 2 opposite cells, but those sisters are still together. We don't separate the sisters until the end of meiosis II. The anaphase 1, the homologs separate, and so we are reducing our chromosome number, but we still have too many copies. I want to get through all these, so I'm going to jump through. We'll come back next time. But telophase 1, we go through what I just drew up there. They separate, the nuclear envelope reforms. The cell may rest in interkinesis. If we're talking about an egg cell, the cell may rest indefinitely. If you're a sperm cell at puberty, they kick in and you go through meiosis 1, meiosis 2, meiosis 1, meiosis 2. And so interkinesis is shorter. DNA replication does not occur in between meiosis I and meiosis II. I said it again. Do not replicate between meiosis I and meiosis II. It doesn't happen. That defeats the purpose of reducing chromosomes. Meiosis II, much simpler. We do the same thing again, but our goal now is just to separate those sister chromatids. Prophase II, chromosomes condense. Metaphase II, chromosomes align. Anaphase II, centromere dissolves, they separate. Telophase II, we reform the nuclear envelope, and cytokinesis occurs.
So,